So, welcome back. Today, I'm going to do a brief little talk on cellular respiration, which is the process that all living things utilize to take the nice complex sugar molecules that were built through photosynthesis or chemosynthesis and break them down into smaller molecules that ultimately lead to us having access to ATP to do cellular work. And so the things we're going to look at today is the process of aerobic versus anaerobic respiration, what's the benefit of one over the other, and how the mitochondria plays a role in aerobic respiration, and specifically the process of oxidative phosphorylation. So those are the topics for today. Hopefully I'll keep it nice and brief and you'll learn something. See you soon. So the major overarching story that we are telling here is the story of entropic manipulation. So when we take this nice, big, complex sugar molecule and we break it down and we end up with a bunch of these little ATPs as well as the trash products of cellular respiration, what we are doing is going from a level of very low entropy in that very highly ordered and complex sugar to a much higher level of entropy with these much simpler, smaller molecules that we are generating. And of course, when we increase entropy in a closed system, we are releasing free energy for doing work. Right? And so that's the story of cellular respiration. It's the opposite entropic manipulations that we saw in photosynthesis. So we are releasing the free energy for work by increasing the amount of entropy in this system by taking complex molecules and reducing them down to much simpler, more entropically favored molecular forms. And so in order to do this, our cells rely upon very specific chemical processes and enzymatic reactions. And in the story of anaerobic or aerobic, right, we are starting with this basic breaking down of the sugar, a step that we see as glycolysis or glycolysis. Right? Lysis as a suffix means to split. Glyco refers to the sugar. So glycolysis is this first step. In aerobic respiration, it's the first step that we are doing to break down the sugar, and anaerobically, it is the step that we are doing to get the ATP. So glycolysis can be done by bacteria, it can be done by humans, it can be done by plants, it can be done by everybody. It's the basic form of cellular respiration. It's the foundation upon which it's all built upon. And now anaerobic organisms who use only glycolysis have adapted more advanced follow-up chemical reactions, such as the process of fermentation, right, to make the best use out of glycolysis that they can. But for the aerobic organisms, it's just the first step along the chain of many different steps that leads to the actual aerobic processes that require oxygen. So anaerobic respiration, it works. It gets the job done. It produces some ATP by breaking down sugar. But aerobic respiration makes a whole lot more ATP. So for anaerobic organisms, okay, they're smaller, simpler things that don't need as much energy. Aerobic respiration is what's required for a complex thing like an animal or a plant who needs more ATP to run their functions. Now, put an animal in an anaerobic condition, right, if we don't have oxygen, our cells will do anaerobic respiration to try to stay alive as long as possible. And so the trash products of that process are what we feel as like soreness in our muscles the days after we work out because our muscles didn't have the oxygen to do the aerobic respiration, so they did anaerobic to get the energy they needed while we were working out. So it works for us, it's just not the best. going to look at glycolysis when I come back. So, little peek at glycolysis. Quick introduction to the process. It occurs within the cytoplasm of the cell. All cells have cytoplasm, including prokaryotic cells. So all cells are capable of doing this step. So this is the first step to all the respiratory processes that we're going to talk about. Okay. And so overarchingly, what this is we're taking glucose, any sugar that we can break down for this process, but glucose is the traditional one to look at, right? And we are ultimately converting it through a chain of reactions into a molecule called pyruvate. And so for each glucose that we break down, we get 2 of these pyruvates. And now for simpler organisms like bacteria, this is cellular respiration. Right? This process is the entirety of what they're doing to generate their ATP. And this process costs 2 ATPs every time that it's run, and it creates 4 at the end of it. So the process of glycolysis only nets a gain of 2 ATPs per glucose molecule. So that's a terrible net. It's not at all productive. And for us, that's not nearly enough ATP. For little bacteria, it works. It's better than nothing, right? It's what they have. And so the way that the whole chain of reactions works, I'm not going to go into the itty-bitty details of it, but effectively, we're breaking this glucose up, okay, by phosphorylating it using ATP. So we are using the phosphate groups from ATP. We're going to phosphorylate different regions of this glucose. Okay? And so in that point, we're manipulating the entropic relationships, right? We're using the breakdown of ATP to shove that phosphate group onto this glucose. That phosphate group makes the glucose uncomfortable. It's not energetically stable anymore. And from there, our chemical systems can manipulate it into changing forms. And so it changes forms a bunch of different times due to this phosphorylation. And ultimately, the last form that we make these chemicals take is these pyruvates here. Okay. And so for bacteria and other things that are doing anaerobic respiration, this pyruvate is trash. And it needs to be recycled back into products that can be used for this process of cellular respiration. Right? And so fermentation is the process of recycling this trash back into something useful. Okay. So that's glycolysis happening in the cytoplasm, taking the sugar molecule, breaking it down, turning it into pyruvates, and processing a couple ATPs out of that. For aerobic organisms such as ourselves, the pyruvate is actually a very useful product that we're about to use to generate a lot more ATP than this measly, stupid little 2 ATPs, which is the next step we're going to talk about when I return. So, set change, right? Obviously, I'm not where I was before. I'm now here in the kitchen where we do learning on Monday and Wednesday. So, as you know, I did distribute a little Google form this morning asking for some feedback about how to perhaps improve the course. And so one of the major feedbacks I've been getting is make the pre-lesson videos shorter. And I agree with that 100%. I'm all the way on board with that. And so I'm going to cut this video off where we are now. Right? When I return to this video, we will be exploring the processes of the Krebs cycle and then the oxidative phosphorylation that actually creates the ATP. At the end of this story, we're going to take a detailed look at the electron transport chain, which is something that was introduced when we talked about photosynthesis, but we will look at its mechanisms a lot more closely in the context of cellular respiration. So yeah, this will be part A of this pre-lesson, and that's it. So cool. Bye.